This is our presentation on the detection of human cystic fibrosis CFTR genetic mutation delta F508 by PCR assay and gel electrophoresis. This is our introduction. Cystic fibrosis is a common autosomal recessive genetic condition that is caused by over 2,000 mutations in the CFTR gene. The CFTR protein is a chloride transporter, and it regulates the ion movement of chloride and sodium ions. When there is a mutation in the CFTR gene, the protein doesn't work correctly and there's abnormalities in water movement and ion transport. And then this causes severe dehydration, which causes mucus buildup, and bacteria buildup, which causes clinical consequences such as lung infections, liver and pancreas dysfunction, and sinus infections. Delta F508 is the mutation that we are specifically looking at, going downwards through the funnel. It is the most common mutation in cystic fibrosis and accounts for almost 70% of cystic fibrosis cases. This mutation is a deletion of 3 nucleotide CTT, and this causes the deletion of an amino acid phenylalanine. And this is a class 2 defect. It inhibits a nucleotide binding domain on the CFTR protein, which doesn't allow the channel to open and function as it regularly would. And then gel electrophoresis and PCR. PCR has 3 steps: denaturing, annealing, and amplifying. In the denaturation step, the hydrogen bonds between the DNA are broken, and in the annealing step, the temperature is raised, so Taq polymerase can amplify the DNA, and then amplification is when the DNA is amplified. Gel electrophoresis is a way to determine that our PCR worked accurately, and the way it works is that there are negative and positive electrodes on each side of the gel electrophoresis, and there is ions in the buffer that help move the DNA. And because DNA is negative, it will move towards the positive end when the voltage is increased. And with that, we will expect a certain band size based on the PCR that we just performed. The purpose of our study was to create primers to successfully detect the delta F508 mutation in cystic fibrosis. The delta F508 mutation is the most common mutation in cystic fibrosis, and it's very important that there is a reliable diagnostic to test for this mutation. And then for the hypothesis, our next figure represents the hypothesis, where the purpose of this figure is to represent a mechanism of our hypothesis. And then, so if we use a wild-type forward primer on wild-type DNA, the base pairs will align accordingly, and it will amplify, creating a PCR product that we will later be able to see in gel electrophoresis. However, if we use a wild-type forward primer on mutant DNA, then the base pairs will not align accordingly, and no amplification will occur. And you can see that where the mutation is when it stops binding to the DNA. This allows selective amplification and will allow us to detect the delta F508 mutation because, for example, if someone has cystic fibrosis and a forward wild-type primer is used on them, then no amplification will occur. However, if they don't have cystic fibrosis and a wild-type primer is used, then amplification will occur at a predicted band size. This next slide is a mechanism for the lambda virus allele-specific PCR. So from the 5' end to the 3' end, this is the DNA sequence for the forward primer, and then this is from the 3' end, from the 5' end, this is the sequence for the reverse primer. And if these successfully anneal, we will get our PCR product, assuming everything else works correctly in the PCR. So the purpose of this figure is to demonstrate how the lambda virus will bind to the DNA that was found on the NCBI database. And then for the methods, we were given these primers, and then we found the corresponding DNA on the NCBI database. And this is exactly how they bind when they go through PCR. And then for findings, if this PCR goes through accurately and we use these primers on the DNA, then we will get amplification in the PCR, and we will see findings in our gel electrophoresis, which we will see in our next figure.
I'm B774 and I will be doing figures 3, 6, and 7. All right, so for what we're looking at here, we have our gel electrophoresis and semi-log plot analysis of our lambda primers. So our main purpose for this was to test our PCR methods, make sure that they work properly, and just have everything in order before we actually test our design primers. So the main methods that we use to do this, we combined our water, our PCR buffer, our DNTPs, forward and reverse primer, DNA template, and our Taq polymerase. So the DNA template was our lambda DNA. And so then once all those were combined, we denatured at 95 degrees Celsius for 45 seconds. We annealed at 55 degrees Celsius for 30 seconds, and then we extended at 72 degrees Celsius for a minute. And then what we were left with was this gel over here. Well, we also ran 5 microliters of our PCR product with 3 microliters of dye to get that gel. Ran that at 125 volts for 30 minutes. And so we got this gel and we got the semi-log plot. And so what the semi-log plot shows us is that based on the distance run by our reference ladder, we were able to calculate that our lambda primers are about 395 base pairs in length, which is exactly what we were hoping for, which proves that this gel worked properly and that our PCR methods do in fact work.
Figure 4 shows the DNA concentration and the purity of sample 1. So on the left-hand side, we have the DNA concentration, and on the right-hand side, we have the purity of the sample. So this sample was measured 3 times, as indicated by the 3 blue bars shown, and the red bar shows the average of those 3 measurements. So for this figure, the average DNA concentration lies right around 255 nanograms per microliter, and the A260 over A280 ratio, or the DNA purity ratio, is about 1.45. And this indicates that there's a moderate protein contamination because the ideal purity ratio is around 1.8. For figure 5, it's set up the same way that figure 4 was set up, except for this figure, we're looking at sample 2 instead of sample 1. For sample 2, the DNA concentration lies right around 54 nanograms per microliter, and the DNA purity ratio is higher than sample 1 at about 1.6. So this indicates to us that sample 2 is more pure than sample 1 because it's closer to that goal of 1.8.
All right, so for figure 6, what we have here is the anticipated binding sites of our published primers, where they should bind, where the mutation is, that's the highlighted spot in yellow, and how that will result in the expected PCR product. So as you can tell, both of the primers, they don't bind directly to the mutation, they just bind around it. They will replicate this about 98 base pair long segment of DNA. And so the main difference that will result is if the mutation is present, then the PCR product will only be 95 base pairs because we'll have that CTT deleted, and that will result in 3 less base pairs, resulting in a 95 base pair sequence versus the wild type, which will have a 98 base pair sequence because it does not have that deletion, and so it will be 98 base pairs in length. So that's what we anticipate from our published primers. All right, so for figure 7, what we're looking at is the results of the PCR and gel electrophoresis of our published primers using our extracted DNA. So the main goal of this was to test our published primers, make sure those work, be able to have a negative control with our extracted DNA, which should be negative, and then also just continue to test our PCR methods to ensure that they work. And so the main methods for this was we combined our water, our PCR buffer, our published primers, forward and reverse, and our Taq polymerase, our DNTPs, and I believe, oh, and our extracted DNA, obviously. And so then we denatured that at 94 degrees Celsius for 60 seconds. We annealed at 64 degrees Celsius for 30 seconds and extended at 72 degrees Celsius for 30 seconds. We repeated that 32 times. And then we got what we see here by adding 3 microliters of our PCR product and 2 microliters of dye. We ran that on the gel for about 30 minutes at 140 volts. And then we were able to take the distance traveled by our reference ladders to analyze exactly how many base pairs our PCR product was. And so based on the semi-log plot, we are able to determine through our analysis that our PCR product was about 98 base pairs in length, which is exactly what we would expect if the wild type DNA is in fact present, which it should have been, all things considered, and based on our previous testing. So, yeah, this just confirms that our published primers worked.
For figure 8, it is a hypothesis test that visually represents how both the wild type and mutant type design primers will bind to their respective DNA. Wild type DNA is not missing the CTT bases at position 508. This would create a band size of 476 in the gel electrophoresis results. For the mutated type primers in DNA, the DNA does not have basis CTT at position 508. The mutated primer was created using the Yaku-Banzhag method, and then the mutated primer will hopefully result into a band length of 473 in the gel electrophoresis results. During gel electrophoresis, we will hopefully see the 3 base pair difference with controls. For figure 9, it is a predicted figure for what we expect the gel electrophoresis results will look like with the design primers. The top of the figure represents the wells and which number they are, and then the left side is the expected base pair length amounts. Well 1 is the 100 base pair plus DNA ladder by SID Labs, which we are using in our lab. Well 3 contains the wild type PCR product, and well 4 contains the mutant type PCR product, and then finally well 7 contains the lambda virus PCR product, which has a predicted band length, or it has a confirmed band length of 395 bases. The lambda and ladder will be used as controls and we'll be comparing them to the design primers to make sure the predicted band lengths of 476 for the wild type PCR product and 473 base pairs for the mutant type PCR product is present and represented. And then finally, in conclusion, each figure and test that we are performing is a piece to the puzzle of creating an assay for the delta F5 mutation for cystic fibrosis patients. The first test is a lambda virus test, which allowed us to test the methods for the PCR cocktail measurements, the PCR thermocycler times and temps, gel creation methods, and gel electrophoresis voltage and times. Next, design primers validated the methods for RSA, while also showing a successful test if someone doesn't have the delta F5 mutation. This was with the 98 base pairs and the gel electrophoresis. This test also validated DNA purification methods because the primer's predicted band length of 98 was present. The next test, DNA purification, allowed us to test DNA without the delta F5 mutation, and this allowed us to test for the delta F5 mutation. And then lastly, the design primers allowed us to create a new method of tests for the delta F508 mutation. And all in all, these primers, these processes and tests allowed us to create a new successful PCR and gel electrophoresis assay.